yes, I would like to welcome our speaker. Sarah, thank you very much for joining us today and willing to share your research and all the interesting things you are doing. So I will start with the short introduction of our speaker. So Sarah O'Connor, she obtained her PhD in organic chemistry in the Massachusetts Institute of Technology in 2001. And then she got awarded with the prestigious fellowship, and she did her postdoctoral time at the Department of Biological Chemistry at Harvard Medical School from 2001 to 2003. Then she accepted assistant and later on also assistant professor position at the chemistry at the Massachusetts Institute of Technology. And from 2011, she joined Johnines Center when she was a project leader and also a professor in the Department of Biological Chemistry. In 2019 was the time that she moved to Germany, and she accepted to be a head of the department at Max Planck Institute of Chemical Ecology. And there she is doing the current research that we will see today. So she has mentored around 13 PhDs and 26 postdocs, which I think is a really big number. In her lab, the research is going into direction to discover new genes which are responsible for biosynthesis of complex natural products. And today she will tell us a bit about some enzymes which might catalyze unusual or not characterized well reactions or the transformations. And she will introduce us to the methods that they are using in the lab. I would like to mention one more thing. Let me just share, because recently we are talking a lot about supporting women in science and promoting women in science. So I think Sarah is a great example of very successful women in science. And I found one thing which I would like to share with you.
I don't know what this is.
Don't worry, nothing bad. Do you see my screen?
Yeah.
Okay. So I found today, looking for some interesting aspect of the woman in science, that some years ago, you were giving an advice to women what is important to succeed and to be a strong woman in science. So I will read it for all the young women. So the advice from Sarah is that my advice for young women in science is to find supportive people, both in life professional and in your personal life. At work, you need supporters to help open doors in science and give you advice when you need it. At home, you need supportive people who are okay with the fact that you spend so much time and energy doing science. So I fully agree with that. I think you need these two components to really find the power and push the things towards. And I think that's why Sarah is very successful. And she was one of the first female director, actually, in Max Planck Institute for Chemical Technology. So that's why I'm very happy that you accept our invitation, and I'm very much looking forward to your talk.
All right. Thank you so much, Monica. And I'm glad you took something away from, I think that was like a sort of a workshop that we did in Gulm, which I'm really glad to hear that at least one person got something from that. So thank you. Thank you. You know, sometimes if one person learns lessons the hard way, maybe they can help other people learn those same lessons an easier way. So anyway, okay. So I'm going to talk to you about plants, and specifically I'm going to talk to you about plant natural products. So Oh, first of all, before I get started, let me thank my research group. This is a picture of us in the early days of the department when we just got started. I really owe all of these people, not just their contributions to the science, but also the contributions to getting the lab up and running again. So I am super, super grateful to all of them. And we have a whole host of collaborators. I'll try to mention them as we go along. Robin Buell, who's really been an outstanding collaborator with all of our bioinformatics and sequencing, Dave Lawson and Jerome Bascan and Elena Conti for supporting us with crystallography. We have some Brazilian collaborators, Ivo, Adriana, and Ana Maria, who help us provide us with plant material as well as chemicals from plants. Vincent Cordevalt and his group for helping us with some enzyme localization experiments. And I'll be talking about some of Benji Lichtman's work. He was a former postdoc and he now has his own group at the University of York. Okay, so as I was saying, I'm a chemist by training and I'm very interested in the chemistry that plants do. And I took this picture of one of my favorite plants, Catharanthus roseus, which I found at the, I think it was the Missouri Botanical Garden. And Catharanthus roseus makes all of these molecules that I've shown here on this slide. And Catharanthus, as well as all other plants, make these complicated molecules, which we call natural products or sometimes specialized metabolites or secondary metabolites, for a whole variety of purposes, for defense, for pigmentation, for interspecies, intraspecies communication. And we're interested in studying these pathways, the pathways that are responsible for the construction of these molecules, for a number of reasons. One reason is that loss and gain of these biosynthetic pathways that are responsible for the construction of these molecules has occurred repeatedly during plant evolution. So natural product biosynthesis, I think, is an amazing system in which we can probe some of the mechanisms of evolutionary processes, and I'll tell you a little example about that. And also, many of these natural products are specialized metabolites. The plants use them for their own purposes. But actually humans can actually hijack those molecules and we use them for our own purposes. So for example, many of them have pharmacological activity. And so understanding the biosynthetic pathways of these molecules can help us think of better ways to produce these molecules and use them for our own purposes. Okay, so let me start by telling you a little bit about one story, which gives one sort of tiny window into the evolution of natural products in plants. And the system that we use for studying the evolution of natural products in the story is using a type of compound called iridoids. Now, iridoids are a type of natural product known as monoterpenes. And for those of you who don't know, monoterpenes are a very widely found class of natural product or specialized metabolite formed from GPP or geranyl pyrophosphate. And in these biosynthetic pathways, a monoterpene synthase will catalyze the cleavage or the loss of this pyrophosphate group to generate this reactive cationic species here that can rearrange in any number of different ways to give what we call canonical or normal or standard monoterpenes. In iridoid biosynthesis, this pathway happens a little bit differently. So we have a dedicated type of monoterpene synthase called geranyl synthase, which generates geranyol, which is then subsequently oxidized to form this highly reactive dialdehyde species. And then a reductase known as iridoid synthase will catalyze the reduction of this system here, conjugated double bond system here. And this will trigger a cyclization to form nepetalactone, which is the framework of all iridoids that you find in nature, this fused 5-6-membered ring structure. And then one of the very simplest iridoids that we find in nature is just the oxidation of this lactol to form the lactone, and this generates the compound nepetalactone. Now, nepetalactones are really interesting in their own right. for their biological activity. It's believed that plants produce these to communicate with insects. And in fact, Mike Burkett and John Pickett showed a number of years ago that different stereoisomers of nepetalactones, when mixed together in different ratios, actually have different repellent activity against different insects. So the hypothesis or the data really suggests that plants actually tune mixtures of these different stereoisomers to sort of tune the biological activity of these nepetalactones. I should also mention nepetalactone is actually the active ingredient in catnip. So catnip is it's a nepeta, it's Nepeta cateria for a cat. And this is one of my cats enjoying one of the cultivars, many cultivars of Nepeta cateria that we have in the lab. And he's smelling the nepetalactone right there. So from a chemical perspective, we were really excited about the fact that there was a system where nature generated these different stereoisomers at these carbons here. And this was a very talented synthetic organic chemist, Nat Sheridan, who really made this observation. And from a chemical perspective, it is very difficult to kind of set and control the stereochemistry at these types of sort of sterically hindered carbons here. And we showed that we could take a species of Nepeta mucini, and just look at different cultivars of this plant, and they had different ratios of these different stereoisomers. So we thought that this was a wonderful system where we could really explore the biosynthetic pathway in this plant and try to come up with the enzymatic and the mechanistic basis for how these different stereoisomers are formed. And so the logical hypothesis was that we knew that an enzyme called iridoid synthase was involved in the formation of nepetalactone, and we assumed that iridoid synthase would, in addition to catalyzing this reduction that I mentioned, would also control the stereochemistry of the subsequent cyclization to set these stereo centers in nepetalactone, and then subsequent oxidase would oxidize it to nepetalactone. And long story short, that did not turn out to be true. And I won't go through all the data in this slide, but basically what Nat showed was that he could take an iridoid synthase from a plant that we knew made only this stereoisomer, and he tested it with all of the Nepeta iridoid synthases that he could find. And he either found enzymes that were not active, or he found enzymes that gave exactly the same chemical profile as what we saw in the Catharanthus roseus that produced only this one stereoisomer. And so iridoid synthase does not control the stereochemistry of the cyclization. And these are GCMS traces, and they're a little bit messy just because we don't just form nepetalactone, but we also form the open form of nepetalactone. So that's what these extra peaks are. And Benji Lichtman, a subsequent postdoc, actually dove into this question, and he, through a series of really elegant experiments, came to a well-supported conclusion that while the enzyme iridoid synthase reduces this oxidized form of geraniol, 8-oxogeraniol, it does not catalyze the cyclization. So iridoid synthase will add a hydride to this double bond system. It will generate an activated intermediate. And then this activated intermediate can spontaneously cyclize to form the nepetalactone structure, primarily that stereochemistry that I showed on the previous slide. And he showed this, one of the experiments he used to show this was to compare the product profile of a spontaneous reaction with an enzymatically catalyzed reaction, and they're basically identical. So that led us to the hypothesis that iridoid synthase, this was a two-part reaction, and that iridoid synthase did the first part of the reaction, and then a subsequent cyclase would trap this activated intermediate and control the stereochemistry of the subsequent nepetalactone. And so how do we go about trying to find these cyclases? And fortunately, nature had given us an advantage in that we knew that nepetalactone production was localized to these oil glands on the surface of the leaf, these trichomes. And so we could get a proteome and a transcriptome of solely the trichomes. And we could look for genes and enzymes that were specifically enriched in this site of where we knew nepetalactones would accumulate. And long story short, this allowed Benji to find a number of genes and corresponding enzymes that were involved in the biosynthesis of these different stereoisomers of nepetalactols and nepetalactones. He found a class of enzyme which he called NEPs, which is a type of it's a short chain alcohol dehydrogenase, and these NEPs will take the reduced form, the iridoid synthase reduced form of 8-oxogeraniol, and control the cyclization into one of 3 stereoisomers. And these are redox type enzymes, and they contain an NAD cofactor, but they're redox neutral. So they've clearly just been hijacked for the cyclization purpose. And then we can find other very similar or identical NEPs that actually are almost identical and sequence and structure, but these are redox active, and these catalyze the oxidation of the nepetalactol to the nepetalactone. And we could show that the expression levels of these different NEPs genes actually corresponded to the chemotype of the cultivar that we were looking at. So that was kind of nice indirect evidence that these genes actually are involved in the biosynthetic pathway. So this is basically how nepetalactones are made. We have an activation of this substrate, and then we have this activated intermediate is then captured by one of these enzymes and cyclized and then oxidized. So with the biosynthetic pathway in hand, we could then try to look at iridoids in an evolutionary context. And so Nepeta is actually a member of the mint family. The mint family is a very large group of plants, otherwise known as the Lamiaceae. And there are a number of subfamilies found in the Lamiaceae, and the Nepetoideae is one of the larger subfamilies. And so what we did was we actually measured with mass spectrometry the presence or absence of iridoids in all, I think it was 53 representative members of the Lamiaceae family. And what we found was that in the Nepetoideae subfamily, there was a complete loss of iridoid production, except in Nepeta, where we observed nepetalactones. And so the logical hypothesis is that iridoids were ancestral in the Lamiaceae, and maybe there were some local losses in certain species, but for the most part it was remained. But there was a major loss of iridoid biosynthesis when the Nepetoideae branched off, and then there was a reemergence or a re-evolution of iridoids, specifically in Nepeta. So we were really interested in asking how iridoids were lost and how they came back. And the first 2 really key observations to make are that iridoid synthase and the corresponding cyclase, the NEPs, are not present in the Nepetoideae outside of Nepeta. So you only see them in Nepeta. You don't see them in any of the other plants in that subfamily. And we took Benji took a very close look at iridoid synthase due to the structure of the gene family. It was a little bit more challenging to look at the NEPs, though Benji is continuing to look at the evolution of the NEPs, but I'll focus here really on the iridoid synthases. It was absolutely clear that iridoid synthase is lost in the Nepetoideae. So that was the key event that led to the loss of iridoid production. And that iridoid synthase really did re-evolve in Nepeta. And I think you can see here in this phylogenetic tree, these are the iridoid synthases that are found in all plants known to make iridoids. They're all very similar across a very, very wide swath of different plant families. Iridoid synthase is, we believe, the ancestor to be a protein known as progesterone beta reductase. This is another type of short chain alcohol dehydrogenase. And you can see that the iridoid synthase from Nepeta, which is way out here, is obviously very different from all of the other iridoid synthases that we know of. So it really seems reasonable to speculate that iridoid synthase from Nepeta actually re-evolved a second time from this progesterone 5-beta reductase ancestor, and just came up with a slightly different variation of iridoid synthase. So basically, we believe that there was a loss of iridoid synthase in the Nepetoideae, and that iridoid synthase then subsequently specifically re-evolved in Nepeta. And Benji looked at this a little bit more carefully. And what he did was he compared the catalytic enzymatic activity of the putative ancestor, progesterone 5-beta reductase, which is believed to reduce a number of different substrates, but one of the substrates it reduces is progesterone, so it catalyzes the reduction of this double bond here. And then he also compared this with the catalytic activity of iridoid synthase, which reduces this double bond here. And he made a series of ancestral sequence reconstructions. There we go. So what he did was he measured the biochemical activity in a quantitative way, so measuring Kcat and Kms, of all of the extant iridoid synthases, as well as the progesterone 5-beta reductases that we found in Nepeta mucini, Nepetalactone, as well as an outgroup, Hysospis officinalis, which is right next to Nepeta in the phylogenetic tree, but it does not produce nepetalactone or any other iridoid. And measured the catalytic activity, comparing iridoid, and this shows the relative activity of iridoid synthase activity compared to progesterone 5-beta reductase activity. And then he constructed 3 ancestors representing what we thought represented sort of the tracking of the evolution of this enzyme along this process. And you can see that what we believe to be, you know, sort of the first ancestor is a multifunctional enzyme, or at least bifunctional. We've only tested it with 2 substrates. So this is a promiscuous type of enzyme. It will reduce both 8-oxogeranial and progesterone. And this is also true for the second ancestor that he constructed as well. But by the time he gets to the third ancestor, you can see that he's generated a sequence that is now more catalytically specific. It has really evolved specificity for the iridoid synthase substrate. So this is really kind of a nice example of an illustration of the hypothesis where a metabolic enzyme kind of starts off a little bit promiscuous, a little bit sloppy, and then when there's some sort of selective pressure for that catalytic activity, it will really specialize and become much more effective for a single dedicated enzymatic reaction. Now, as we were doing this, thanks to our collaborator, Robin Buell, who had sequenced the genomes of a couple of species of Nepeta, we actually realized that the iridoid biosynthetic genes were clustered on the genome. And I'm sure, as many of you know, this sometimes happens in plants, where you kind of get a co-localization or an enrichment of genes from a biosynthetic pathway on this approximately the same location of the genome. This is not an operon. I mean, these are not. They all have their own promoter, and I mean, it's not as if they're, it's not an operon like in a bacteria, but they are more or less contiguous on the same place in the genome. And so I'm just showing you kind of a schematic of them here from Nepeta mucini, as Nepetalactone is a tetraploid, so that's why you see 2 versions of it. But you see the iridoid synthase along with the neps, along with another cyclase-like protein, which I didn't really mention, and geraniol synthase, one of the upstream enzymes. So this was kind of interesting to us that, you know, this nepetalactone biosynthesis is actually an example where the genes are clustered on the chromosome. And the phenomenon of gene clustering in plants is very poorly understood, and I'm not really going to claim to shed much light on this. But one question that we wanted to ask using the nepetalactones as a system is what is the timing of gene cluster formation relative to product formation? So what happened first? Did the enzyme activity evolve first, and then the cluster formed, or did, you know, these genes happen to be located contiguous to each other on the chromosome, and then the enzyme evolution occurred? And so Benji could actually use his analysis to really try to address this question. And so just to remind you, this is what we call the gene cluster locus. So this is where the extant iridoid synthase is. It's clustered with the neps and the geraniol synthase and so forth. But what he also noticed was a locus where the progesterone 5-beta reductase is. This is the alleged, you know, extant version of the ancestor protein. And when he looked very closely at the transcriptome in Nepeta, or the genome in Nepeta mucini, he saw what we call a silent iridoid synthase. So this was a totally, he could clone it out of the genome. It was not expressed, but it was a perfectly functional gene on the genome. And when he cloned it out, it was perfectly active. It had basically the same catalytic constants as the iridoid synthases that were very highly expressed in Nepeta mucini. But this silent iridoid synthase was actually not located in the gene cluster. It was located next to progesterone 5-beta reductase. So we kind of viewed the silent iridoid synthase as kind of like a fossil. So the kind of the hypothesis that we have for how this happened was that at our progesterone 5-beta reductase locus, the progesterone 5-beta reductase duplicated and duplicated to generate Benji's reconstructed ancestors 1 and 2, these kind of multifunctional, not so specific enzymes that could catalyze both progesterone reduction and iridoid synthase through this tandem duplication event. And then after the second ancestor, there was another tandem duplication event. And this is where one of these duplicates underwent, we suspect, positive selection to subfunctionalize, to be highly specific for iridoid synthase activity. And this is ancestor 3. This is Benji's ancestor 3, we speculate. And then somehow, ancestor 3 probably duplicated again, and then somehow was recruited into this ancestral cluster containing the neps and the geraniol synthase and so forth. We don't know the mechanism for how this happened. We call it dispersed duplication. We don't really know what happened, if there was a transposable element or something like this. We can't really speculate too much on this. But this old ancestor 3 remained next to its, ancestor progesterone 5-beta reductase, and Nepeta mucini, it was just silenced. In Nepeta cateria, we could actually see a partially pseudogenized form of iridoid synthase next to progesterone 5-beta reductase. So this has a number of truncations in the middle of it. So we assume that this gene is in the process of sort of being destroyed and taken out of the genome. And then on the other chromosome of Nepeta cateria, we saw a complete loss of iridoid synthase next to progesterone 5-beta reductase. So this is kind of a nice example of how, you can look at the catalytic activity of these ancestor and extant proteins, and then you can kind of look for little clues of where they might have appeared on the genome, and you can try to make some predictions and put together a story of how these proteins might have moved around to form a gene cluster. But basically, the location of the iridoid synthase pseudogene, that silent iridoid synthase, really suggests to us that gene function, the enzyme function, preceded the formation of the cluster. So we think that plants were making nepetalactone and iridoids before this gene cluster was actually formed. And of course, the question is, okay, so we have a little bit of the answer of how iridoids were lost and re-evolved in Nepeta. The other question is, why did this happen? And I think it's important to point out that Nepeta does not occupy any different environment compared to the rest of the Nepetoideae. So I can't really think of any, specialized environmental or, predator or herbivore that would induce formation of nepetalactone. But one thing that we do speculate is that the genetic context of Nepeta, specifically the presence of these helper cyclases, these NEPs, may have helped driven the specialization of iridoid synthase. So Nepeta, for whatever reason, had the ancestor of the NEPs. We think it's probably an oxidoreductase that was involved in making a type of monoterpene that's found only in Nepeta, and that there was coevolution between NEPs and iridoid synthase, that the fact that we had, that the Nepeta had this genetic background with this enzyme that might have helped cyclization, this may have really helped drive the specialization of iridoid synthase in some type of coevolution event. Okay, so that was kind of a story about evolution in plants, and I wanted to now move to a different natural product pathway that we work on to kind of give you an illustration of how we identify mechanisms that plants use to generate chemical diversity. And the pathway that I want to highlight for this is a pathway involved in making an anti-cancer agent in this plant, Catharanthus roseus. And specifically, I'm only showing about 2/3 of the pathway on this slide. But specifically, I want to highlight this section of this pathway shown here, which is really making kind of the key scaffolds, these molecules known as catharanthine and tibercinine. Now, it has been proposed that the substrate, the starting material for tibercinine and catharanthine is this molecule here called dihydrocecidine. And in fact, you can imagine even a number of different scaffolds that are formed from this starting material dihydrocecidine if you go to other plants. And we've also combined this work with another plant called iboga, which makes this compound here called coronarity. And so we see tibercinine in both catharanthus and tabernanthe iboga. We see catharanthine only in catharanthus, and we see minus this coronarity only in iboga. And we care about these molecules because they're so structurally interesting, but we also care about them because in this case, they have really important pharmacological activity. So as I mentioned, catharanthine, which I'm showing in red, and a derivatized version of tibercinine shown in blue, dimerized together to form vinblastine, which is an anti-cancer agent. And that coronarity, again, a slightly modified version of it, forms the basis of a molecule that's produced in iboga or tabernanthe iboga known as ibogaine. And this has really interesting anti-addiction activity. It was actually a guy in the 1960s heard about this molecule that was used by a variety of tribes, mostly in Gabon, in religious ceremonies. It produces hallucinations, and he wanted to try it because that's what he was into. And he was also addicted to heroin at the same time. And he took this molecule, and he noticed that after he came down from the trip of ibogaine, that he didn't really have any cravings for heroin. And he stopped taking heroin, and he had no withdrawal symptoms. And then he went and he said, okay, let me test this. And he got himself addicted to heroin again, and then took ibogaine again. And the effect was the same. He killed his withdrawal. He mitigated all the withdrawal symptoms. So this has real promise as an anti-addiction agent. It also has some pretty severe cardiac side effects. So there's been some issues with getting this into the clinic, though there's a lot of interest in making analogs of ibogaine that might have mitigated or much diminished side effects, so it can be used. So very interesting molecule. Okay, so I mean, how do you go about figuring out how these molecules are made, and where does dihydrocecidine come from anyway? And so just to kind of give you a little bit of background, I'm not really going to go into the chemistry in this talk, but I mean, basically, we knew this molecule here, stamagine, because it's stable and it had been isolated from many, many plants and had long been hypothesized to be a biosynthetic precursor for vinblastine and these types of natural products. And it was a really amazing organic chemist in the 1960s, Ernst Wenkert, who had actually proposed the structure of dihydrocecidine. He had proposed that was actually the precursor for those catharanthine, tibercinine, and coronarity molecules that I showed on the previous slide. So what we had to do was to try to imagine a way that stamagine could be converted into dihydrocecidine, and to try to find the enzymes that would carry out those steps that we could, saw in our imagination. And this is what we did. And basically what happens is that stamagine is acetylated, and this is a very common modification that plants do to try to stabilize molecules. So they'll always be putting acyl groups on hydroxyl groups to stabilize molecules. And then we realized that stamagine could be readily oxidized by this flavin-containing oxidase that we found here. And this carbon-nitrogen bond will be oxidized. And then a reductase will actually reduce this oxidized version of this molecule. And this reduced version, exactly as Wenkert had predicted, will basically fall apart to form dihydrocecidine. And Lorenzo Caputi and Jakob Franke in my lab actually managed to isolate not dehydroxycodine itself, but a trapped form of it, and actually could structurally characterize it and show that it was in fact the substrate for the downstream enzymes. So we really had kind of nice proof that in fact dehydroxycodine is in fact, you know, the right end product that we've been looking for. They named it angrelene because it made them angry to isolate. So, okay, so that's how we got to dehydroxycodine, in part thanks to our work in the enzymology and Ernst Wenkert's amazing chemical insights. And what Ernst Wenkert had predicted was that dehydroxycodine could undergo any one of a number of cycloaddition reactions, such as Diels-Alder reactions, to form carbon-carbon bonds in slightly different ways to lead to these three different structures. And using a whole lot of RNA-seq data, and by analyzing that RNA-seq data using co-expression analysis, so we looked for genes that were co-expressed for those with those genes that I showed on the previous slide. We found these hydrolases. They were annotated as hydrolases, and they're homologs of hydrolases that catalyzed the formation of dehydroxycodine, or the trapped form of dehydroxycodine that we could isolate into these three different structures. And so I think you can imagine, I just showed for people in the audience who were interested, if the double bonds are arranged in this way, you can imagine the bonds forming in this way. I've tried to color code it in red. Conversely, if you kind of orient the molecule a little bit differently, it's exactly the same molecule, but you orient it a little bit differently, and now you react these bonds together, you're going to form these bonds that I've highlighted in blue, which will give you catharanthine. And then we hypothesized that coronaridine, which is exactly the same as catharanthine, except this double bond is reduced, and it's the opposite enantiomeric configuration. So they're the mirror image of each other. And what we predicted was that there would be an enzyme that would just catalyze the enantiomer of catharanthine and then subsequently reduce this double bond. So we were amazed. We really wanted to see how these enzymes actually work. So we were able to crystallize all three of these enzymes, and we could understand a little bit better how they worked. So for catharanthine synthase, we could actually trap something in the active site, and this showed us that this enzyme actually works via a two-step reaction, proceeding through this reaction intermediate. So this was actually trapped in the active site of the enzyme. And we can see that here. And we can actually make a whole variety of mutants that I've tried to highlight here in yellow that modulate the product selectivity of this enzyme. So we can make a whole variety of mutants of this enzyme that make the other cyclase products, such as tibersenine. With tibersenine synthase, as I mentioned, the molecule has to be completely differently oriented. I show here the substrate docked into the enzyme active site, and you may be able to see that it's in a completely different conformation with these double bonds parallel with each other, totally different from in the previous slide. So really what we think these enzyme active sites are doing are playing a big role in changing the shape of this highly flexible substrate and binding it in different ways that will facilitate that cyclization to happen in different ways. Now, for the last product, for coronaridine synthase, it was a little bit trickier because, in fact, we were never actually able to isolate catharanthine in this reaction. Instead, what happens is that this enzyme, coronaridine synthase, goes through this highly reactive intermediate that we propose as this intermediate here, which is not very stable on its own, but it can be trapped by a reductase that actually acts upstream. So it's recruited to work downstream. So this proceeds via a completely different cyclization mechanism. And again, we can see here coronaridine synthase, and we can make mutations to these regions as well to sort of change the product selectivity. So we've got one substrate, we've got three different products. formed by 3 different enzymes that are over 70% amino acid identity. And the way that these enzymes control the product specificity is of the orientation of the starting substrate, specifically the orientation of this part of the molecule relative to this part of the molecule, the green versus purple parts of the molecule. They have slightly different mechanisms depending on the product. One catalyzes what's called a concerted reaction or a Diels-Alder reaction, whereas 2 of the others catalyze a stepwise reaction, which is called a monic reaction. And we believe that a big part of this is involved, the cyclases can tautomerize or slightly change the position of double bonds in this ring. And that opens up lots of possibilities for different chemistry with this highly reactive substrate. Okay, so I think that's for us at least, that was a really nice example of how nature can take, sort of one protein and elaborate on it to generate 3 different products from the same substrate. And kind of with that knowledge in hand, we wanted to really think about whether we could put any of these genes to use. And of course, I'm sure you all know, with the advances in synthetic biology, we can now think about taking genes from plants, maybe plants that are hard to grow or plants that only produce these compounds in small amounts. We can put them in a simpler host like yeast or bacteria or plants like tobacco that are a lot easier to grow and try to overproduce some of these complicated plant molecules more easily. And so we've been doing this in a plant called Nicotiana benthamiana, which is a type of tobacco. Maybe some of you work with it. It's a very easy to transform plant, and we can do a transient transformation where we can take all of the enzymes from a starting substrate from the kind of mid-region of the pathway, and we can co-infiltrate this substrate along with these genes here. And we can actually form the final product, tuberosine. And one of the wonderful things about these types of synthetic biology approaches is you're not limited to the natural compound. We can also think about making new to nature or unnatural products. So for example, we can make fluorinated versions of these compounds by feeding in, instead of tryptamine, a fluorinated version of a tryptamine. And then we can incubate these with the downstream enzymes, and we can isolate fluorinated alkaloids as well. So, and again, I mean, the plants have evolved these compounds to serve their own purposes in defense or signaling and other things that we could imagine. Plants did not evolve these alkaloid natural products to cure human cancer. So you could imagine that if you tweak the structures a little bit, you could drastically improve the pharmacological properties, things that plants would not be optimizing for. So we're very excited about trying to be able to use some of the biosynthetic machinery from these plants to be able to make interesting new types of molecules. Okay, so I think I'm going to stop there. And thank you again for the invitation. And I hope I left time for questions. So I'd be happy to try to answer any questions. Thank you.
Thank you very much, Sarah. It was really nice presentation. I'm really impressed. Before we start the discussion, I just want to mention that we will still have today literature seminar, so the journal club. So please, students who sign for that, stay after, so don't disconnect, because we stay in the same link. Okay, I think we can now open the discussion. If there are any questions, please raise your hand or just start. Okay, maybe Kyle, you can start.
I think I beat Salim by like a nanosecond there. Sorry, Salim. Hi, Sarah. Grok. I saw your talk also in Bielefeld a couple of weeks ago. So
that's
sorry we didn't.
Of course, that's right. Yeah.
Yeah, sorry we didn't get to chat. I noticed you took off right after your talk, or the first day, so we didn't get to chat on the next day. But it was great to see and very inspirational work. I'm just kind of curious about your ideas for synbio applications in the future, because I bet. Because I mean, there's such complicated pathways, right? And you're talking about products of many different sort of chemical structures that have perhaps differences in hydrophobicity, hydrophilicity. How practical is to bring these pathways into something like yeast or even algae or something like that? So is compartmentalization going to be an issue? Is it better maybe to mix and match different organisms that have 5 different parts of the pathway to then have the interplay between the hydrophobicity, hydrophilicity? I'm just curious about your discussion on that point.
Can I just say that I would love it if somebody tried to reconstitute this pathway in algae or cyanobacteria. I think that it could be a really interesting host. So E. coli is really challenging because a lot of the eukaryotic proteins just don't do well in E. coli, especially the membrane-bound P450s. It's hit or miss whether they express well in E. coli. In terms of yeast, there are definitely people trying to reconstitute vinblastine in yeast, and there's been some success. Not in vinblastine, but in some of the upstream intermediates. I mean, we've done a little bit. There are other people who've done a much more thorough and successful job. The titers are low, and nobody really has a great explanation for that. The 2 things that I think are problems are just problems with protein expression and protein stability. And also, some of these intermediates are very highly reactive. So one of the things that we're not really chemical engineers, but I like talking to chemical engineers to think about how, biochemistry and biophysics can kind of inform chemical engineering or metabolic engineering. And we're really interested in how these pathways are actually physically organized. And I didn't really talk about this data because it's still pretty early, but I mean, a student in the group has gotten crystal structures for this vinblastine pathway of all of the downstream enzymes except for the P450s, and has tried to model together interactions for how these proteins might be interacting with each other. And we're really trying to build a, kind of an accurate in planta model of how they might be organized. And so she's set up some really nice fluorescence assays in the lab where you can transform into proteins in a benthamyana leaf, and with luciferase tags, and then measure the luminescence if they interact. So we're trying to put together a picture for how the protein sort of superstructure might look like, and hopefully that might provide some clues and ideas for how to do better engineering in heterologous systems.
Sure, yeah. I know with forskolin that there's a single chaperone that if you express it with the pathway, it brings everything together and kind of locks it in. And it goes from, tens of milligrams per liter to grams per liter almost instantly in the heterologous system. So it's great that you're doing that kind of added level of structural biology. I'm sure it will really help. If you want to express anything in algae, send us a mail. We'll be happy to do it for you. So
okay.
We're very, very successful at doing mono, di, and sesquiterpenes in algae.
Okay. And that's another problem with yeast, because these are monoterpene based, and yeast isn't great at making monoterpenes. So
what we've learned is that a lot of monoterpenes have an apoptotic stimulation effect in microbial cells. So it's true for a lot of monoterpene products in clammy with linalool, for example. If you try and produce limonene or linalool, the cell dies after about 10 milligrams per liter just because it turns on apoptosis. So it could be that the early intermediates may be toxic to your microbial systems, but we can of course test that for you. Geraniol is pretty straightforward for clammy. So let us know.
Yeah. Thanks.
Thanks very much.
Okay, thank you very much. Maybe now Salim.
Thank you very much, Sarah, for this excellent talk. I have a question about the evolution of the gene clusters. Do you usually find intermediates of them? I mean, you are assuming that the enzyme, the genes have already the function, and then they got together.
Right.
Do you find usually intermediates of these gene clusters, and why we don't see them in animal systems?
So sorry, intermediates of gene clusters.
Yeah, I mean, if you have, if a pathway needs 5 genes, do you find a cluster of 3 genes and cluster of 4
So
genes?
actually, in plants, usually that's the case. It's usually a partial gene cluster. I mean, like for all of the many of the plant pathways that are reported to be clustered, it's part of the pathway. And that's true for the nepetalactones as well. Not every single gene involved in nepetalactone biosynthesis is included in the cluster. I mean, I think there are a couple of very short pathways that are clustered and all the genes are included. But I mean, like for, noscapine, which is a very long alkaloid pathway, and Ian Graham at York figured this out, that it was clustered. It's like the last, half of the pathway is clustered. As to why it is, I think probably the best answer is, and this was kind of proposed by Ann Osborne, and I think she's probably correct, is that if you have the genes in the same location in the genome, it's easier to co-regulate them.
True. Yes.
Because of all sorts of genomic mechanisms. There's been a hypothesis that pathways with toxic intermediates are more likely to be clustered because it's more important for the co-regulation to happen. I don't know that I believe that. I don't, I think there are plenty of pathways with toxic intermediates where the genes aren't clustered. So it's probably one of many possible reasons for driving the clustering of plants. I mean, I don't really know why some pathways are clustered and others aren't. I do think that plants need time to cluster. I am kind of convinced that, it's something that clustering happens slowly over time. So maybe younger pathways are less likely to be clustered than older pathways, but there are lots of evolutionarily old pathways that are not clustered.
And do you see a correlation between the production of geranyl and the formation of the neptune of the final product?
Yeah.
I mean, if the precursors are more available?
So it's a good question. We have not measured levels of all the intermediates in the plant, which is tough to do because they don't accumulate, and it's kind of tough to measure. These things they're small and they can be tough to measure. I can tell you that the genes are co-expressed. I can also tell you in heterologous systems, if you want to interpret, some of the metabolic engineering efforts, relate it back to the biology. If by overexpressing like DXS, one of the early, rate limiting steps, you really ramp up production of the final product. So yeah, increasing precursor availability, at least in heterologous systems, really helps.
Thank you very much.
Okay, now Nicola, you can ask.
Okay, so part of my question has already been asked to you by the previous speaker. So I will reformulate a bit, and
okay.
My actual question is that, so you were able to isolate these genes from this other plant, and you were able to express them in Nicotiana, right? Do you think a mechanism like this is feasible to have a substantial production of the final product? Because like there are many, like lately in these years, I think there was a paper that was looking at it, and it's dated on I think it was quite recent, 2019 or something like that. They were able to produce the whole pathway of cannabinoids inside yeast. And this was actually very interesting, as like you were able to transport all the synthetic pathway from plants into yeast and having actually apparently a great production, a great conversion. Do you think this is still possible in Nicotiana to obtain such like a great production, or is better to like aim for something different?
I think it depends on the product. I mean, we've struggled with, so it could be that somebody's going to get some of these compounds to produce very well in yeast. There are certain classes of compounds that are not so easy to produce in yeast, and monoterpenes are one of them. And so the first thing that you have to do in engineering yeast is yeast doesn't make monoterpenes. So you have to try to engineer out the farnesyl pyrophosphate synthase because it yeast immediately just tries to go and make sesquiterpenes and triterpenes, so C15 and C30. It really doesn't like to stop at C10. So you kind of have to try to divert some of the flux from the sesqui and the triterpenes into an artificial monoterpene without taking too much out, because then you kill all the sterol production in the yeast, and the yeast are a lot more fragile and they die. So that's like a huge hurdle of making monoterpenes in yeast, because you're really messing around with the fundamentals of the sterols that make yeast survive. So for that being said, I mean, people have managed to get monoterpenes and monoterpene indole alkaloids produced in yeast. And there are also tricks that you can think of. Like, I mean, if you're willing to feed a precursor to yeast, like you could feed nepetalactone or geraniol to yeast and have yeast convert from that, and that's actually a pretty reasonable idea. And that works pretty well. Yeah, I get asked, is Nicotiana a good system? I mean, to be honest, one of the main reasons we use it is as just like a small scale testing tool. And it is so easy and convenient to use it to just build pathways really, really quickly. It's a transient transformation. It's modular. It's easy. We can get the answer in a week. And, like if we want to test unnatural substrates, to see if unnatural substrates are turned over by the enzyme, it's just really easy and super fast to do. And one thing that we can now do, which I didn't show in this talk, is one of my students figured out you can transform in the proteins into the Nicotiana leaf, and then he takes like a hole puncher, like a paper hole puncher, and he punches out these leaf discs, and then he puts these in I think a 24 well plate with little bits of buffer containing different substrates. And the substrates will diffuse into the leaf disc. So we can actually do like this very small scale assay of substrates, on this leaf disc. And it's just, no, you're not going to scale up leaf disc, but it's really hugely convenient to use in the lab to get really quick answers about enzyme activity.
Okay, thank you.
Jagis?
Okay, hello, Sarah. I was able to catch your talk towards the very end. I'm sorry. The question is about the common knowledge about the pathways, the clusters that you mentioned. So, how likely for a cluster that is normally responsible for example, detoxification of xenobiotics, to be also responsible for the metabolism of the native metabolites that is already found in the plant? Is there any common knowledge about this?
It's an interesting question. If there is, I don't know it. But like in mammalian systems, you have detoxification enzymes that are really non-specific. So like mammalian P450s are really known for having these open active sites where you can get these xenobiotics and different molecules binding in all sorts of different orientations, just getting hydroxylated just so that you can get them out and get them to the liver, excreted and so forth. Plant, and this is sort of my subjective opinion, but I mean, plant enzymes involved in metabolism tend to be really specific. I mean, so like the P450s that we work with, I mean, they'll work with two substrates, two nearly identical substrates, and that's it. So, I mean, the idea of, this idea of just, sort of detoxifying xenobiotics by, like a P450 that might be involved in the pathway, that's generally not the case. I mean, there are like glutathione transferases and so forth that are pretty in plants that are pretty good at, adding glutathiones to things. You do have, some non-specific redox enzymes, that'll oxidize things, alcohols to carboxylic acids and so forth. But generally speaking, like the metabolic pathways aren't generally involved in that as best as we know. But it's an interesting question. I don't think we have a complete answer to it, but
All right, it's just from a perspective, if the metabolic structures are very similar, let's say you have a benzene ring and you have a bunch of like a carbon structure, if they are very similar, maybe they can actually overlap. That was my thinking.
Yeah.
I don't know if you have come across with something similar, but probably not. It
Yeah.
appears to be. All right, thanks for the answer.
So it's really interesting to think about. Yeah.
All right.
Okay, I have my question. It's more like the philosophical question. If iridoids were, or they are not essential, and Planck decide to get rid of them, why would evolution introduce it again? What do you think?
Yeah, so I mean, my initial goal was to make a species of nepeta with no nepetalactone in it, and it happened. And nepeta is proving to be very painful to transform. So we're actually kind of looking for other plant systems that are a little bit more genetically tractable, where we can try to answer like a similar type of question. Yeah.
Okay. Kyle, I will just ask another one. It's more technical because you said you did proteomic and transcriptomic, extracting only material from the trichomes. How did you do that technically?
It's a dry ice abrasion, and you have to do it on a case-by-case basis for plants, because I mean, it worked really well for Cateria and Mussini, Nepeta Cateria and Nepeta Mussini. And then there's another Nepeta species, Sibiricus, which we absolutely could not manage to get the trichomes off of. So if it works, it's pretty trivial. You know, it was a pretty standard protocol that we used. I mean, but it was like a dry ice abrasion. You just basically shake it in pulverized dry ice.
Yeah. Okay, thank you. Kyle, again.
So it's actually a two-part question comment, technical as well. So you mentioned Ian at York, Ian Graham. His mutants in poppy, I think, were done by chemical mutagenesis, right? And they've made all of the different opioid variants just by knocking out things and then high throughput screening. Could you apply that technique to make your non-iridoid producing plant?
Yes, yeah, you could. Yeah, it would just take a while. And I think that Ian was, they're a little bit lucky in the opium poppy system, because there are so many naturally occurring cultivars with different alkaloid profiles. And I mean, that was a great starting point for them. Whereas, I mean, it's a little bit, they could use some natural variation as well. But yes, we could absolutely do that. Yeah.
And a second follow-up point to the trichome removal. Have you tried using pressurized fluorocarbons?
Nope.
So there's a patent. So we've been looking at fluorocarbons for extraction of isoprenoids from microbes, and we just and it's working very well. We submitted a patent on blah blah blah. We can talk about it more later, but in our research, we found that one of the ways that cannabis oil is made is through this pressurized system using fluorocarbon gases. So it effectively operates by chemically removing all of the trichomes from the cannabinoid plants, and then you have kind of a native state extract of isoprenoids when it's done. So if you want, I can look up the company that makes this. Though it's a German company, I think. Okay. And they have this system. It's like a box, and you put your plant tissue in it, and it sprays it under pressure with fluorocarbon liquids, and it effectively dry cleans the trichomes right out of the plant. So,
wow. I mean, and I know probably your RNA would not survive that, but you could probably get a proteome from it. I'm sure the proteins would survive.
So it's used for isoprenoids almost exclusively, as far as I know. But yeah. Question mark. I think the plant tissue is destroyed by the end for sure. Yeah.
Oh wow, that's really cool. Yeah.
Yeah. I'll get your email from Monica and then send you the info. Yeah.
Okay, that'd be great. Thank you.
Okay. Do we have more questions from someone?
I think there's a question about psychometrella.
Yes, in the chat.
Yeah, and the answer is that's a great question. We have not tried it, and the reason why is because with the tobacco leaf, the advantage of the plant expression system from our perspective is just the speed. We're really trying to do things quickly, you know, to just mix and match different combinations, get really quick assays, not use that much substrate. With psychometrella, even though it's got this, you know, relatively efficient homologous recombination, you've still really, it's a stable transformation, and it's just time consuming. So I agree, it's interesting to try if, you know, you're interested in developing, you know, a bioreactor, stable bioreactor kind of situation. But for our purposes, at least for right now, it's kind of the speed that we're going for.
Okay. Thank you very much. So I think with that, we can finish the lecture time. Thank you very much, Sarah, again, for your time and really beautiful research you do when you share.
Thank you.
Thank you very much, everyone who joined and also was active. So now, please, people who join for the journal club, stay connected, and the rest can go. Sarah, do you want to have a break, 5 minutes or something, or you are fine to?
That would be great. Let me get a cup of tea, and then, yeah. So,
okay, so
5 minutes would be wonderful.
15 past.
That sounds good. Okay. All right.
Okay.
Great. Okay. See you in 5 minutes.